How do enzymes bring about such high rates of chemical conversions? Enzymes eventually bring down this energy barrier, making the transition of substrate to product more easy. Concept of activation energy on x-axis tells progress of reaction, on y-axis is potential energy. Substrate at higher energy level. Then there is transition state. Now activation energy without enzyme. The activation energy with enzyme is smaller than with the one without, and then product. So y-axis represent potential energy, x-axis represent progression of the structural transformation or states through which the transition state. All other intermediate structural states are unstable. Now the nature of enzyme action. First, formation of enzyme plus substrate, then enzyme substrate formation into enzyme product and enzyme plus products. So first the substrate binds to the active site of the enzyme, fitting into the active site. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. And last step, the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Now, this was the catalytic cycle of an enzyme action. Enzyme substrate, then enzyme substrate complex, then enzyme product complex, then enzyme plus product. Short-lived and dissociate into its product. Unchanged enzyme. The factors affecting enzyme activity includes temperature and pH, concentration of substrate and competitive inhibitor. Optimum temperature and optimum pH.